So the first lecturer is Dennis Wang, who's a professor both at Imperial and in the National University of Singapore, and has a long history working on longitudinal modeling in population data. So over to you, Dennis.
Thanks, Magnus, for the warm introduction. And yeah, I haven't worked too long on this longitudinal data or population health. I kind of got into it during the pandemic, like most people get into new research questions, but that was an excellent time to care about public health. So welcome back, everyone. So I know after lunch, you may be a little sleepy-eyed. So you'll be glad to know that this will be a very kind of light talk. So I'm not going to ask you to install any packages or do any coding. You can just sit back. The only interactive thing I'll ask from you is just raising your hand. Okay, so not like Elon Musk raising your hand, just a regular raise of hand. I'll be taking some polls, and also if you have any questions, do raise your hand. I stitched this slide together, copy and pasting from some various publications. So the colors can be confusing. So just stop me if it's confusing. I'm sorry I didn't have time to kind of redraw some of the figures. So this talk, I guess what I want you guys to get out of this talk is just some awareness of possible applications of machine learning methods on population data, and kind of specifically kind of longitudinal data or time series data coming from population cohorts. And I'll define that with a few examples from both UK and Singapore. Well, let's see how this laptop may be frozen. I should test this before. Okay, at least the mouse pad works. Okay. So first, before doing modeling, we have to kind of test your assumptions. Okay, so we're gonna ask you to assess how healthy is the human population. Okay, so here's your first quiz question. Okay, so there are roughly 8 billion people in the world today. Okay, so which distribution represents the population geographically across continents? Okay, so let's just put up your hand. So how many people think it's A? A, okay, that's a decent number. Yeah. What about B? Pretty simple, similar. C? Oh, so it's pretty almost. It's quite a yes, split decision here. So the examples are quite similar, but it's actually B is correct. Okay. All right. So I think people tend to overestimate a little bit the number of people in Africa and in North America, right? But so this is kind of the rough distribution. So each individual represents roughly 1 billion people. Okay, so I'll go back to this example. Reason why I'm showing this will become apparent later. Okay, so next question. Okay, so don't be afraid to guess. Okay, there's no marks. This is just a formative assessment. So according to the United Nations, not United States, I gotta be careful of that. According to the United Nations, the world population will increase by another 2.4 billion. Okay, so that's interesting. So it's still projected to increase by 2.4 billion, despite low fertility rates in some countries. Okay, so the main reason is that essentially which age group will most of that 2.4 billion belong to? Okay, so how many people think it's children? Raise up your hand. Okay, adults like us. Okay, and not many people voting. So very old people. So rest of you guys, very old people. Yeah, that's interesting, right? So it's not very old people or children. It's actually adults. Okay, So this is kind of, I think some of the bias in the media about, aging society and so forth. But you have to remember, there are still people being born, okay? So it's not like, all fertility has stopped and we only have old people later on. So actually the bulk of the population will be adults, okay? Which is quite interesting for kind of research, because a lot of the health research is focused on very old and children, okay? But this group, it's quite a large age range, right? 15 to 74. That's kind of known as actually the economic drivers of the world, right? That's the working adult population, right? And how will kind of health impact their productivity, their impact on society is becoming more important. Okay? But don't feel bad for answering incorrectly, right? So only 36% of the people chose adults. So next question, this is a bit more health related. So there's a lot of vaccinations now. So how many of the world's 1-year-old children were vaccinated against some disease in 2019? So this is before pandemic, right? I mean, a lot of people got more vaccines during the pandemic, but before the pandemic, yeah. How many people think it's 20%? Less than 20%, okay. How many people think it's around 50% of children or 1-year-olds? Okay, that seems like a majority. And how many people think it's more than 80%? Okay, so few are pretty optimistic. And you should be optimistic. It is more than 80% of the children, 1-year-olds, have been vaccinated. Okay, so there's actually tremendous progress in the last 50 years on developing vaccines, but also giving out vaccines, making it more accessible. Okay, and what you also see, like especially in developing countries, that the rate of kind of infectious diseases, really deadly infectious diseases, have dropped. Okay, and so this kind of relates to the next couple of questions. So in lower income countries, then back then in 1970. The life expectancy was 44 years. What do you think it is now? Okay, how many people say A, 40 years? So remain the same. Okay, B, 50 years? Okay, one. C, 60 years. Okay, great. You guys are learning to become optimistic. So yeah, you're in the minority that gets right. So it's actually the life expectancy, even in lower income countries, have increased. And one of the reasons for that is because we kind of prevent a lot of infectious diseases, right? That caused people to die earlier in those lower income countries. Okay, so what about kind of more complex conditions like depression? Okay, so I think most of you have heard of kind of this new kind of mental health pandemic going around. So what happened to the global suicide rates in the last 20 years, do you think? It decreased. How many people think it decreased by 25%? No one? Okay. B, stayed about the same. Okay, stayed about the same, right? Or C, increased by 25%. Okay, so it looks like majority, right? So unfortunately, you guys fell for my misdirection there. So it has actually decreased by 25%. Now remember though, suicide is actually, if you commit suicide, it's kind of like a very severe form, right, of mental health condition or depression, right? But I think what's becoming more prevalent is kind of more subtle signs, right? Different forms of mental health or mental well-being conditions being kind of more apparent, right? So this I guess from in terms of like machine learning problems, right? You have to realize that essentially means there's less labels of severe cases of mental health to potentially train. Okay, so it's going to be if you're like working on kind of mental health there may be like it's been more difficult to label or phenotype some of the conditions. So it's not saying that mental health isn't a problem, but it's just that, you know, we're kind of becoming more aware of different forms of mental health. So a little bit something, since this is ELLIS Summer School, so something a bit more European centric. So in Germany, so this is now looking at the kind of distribution of the population by age. Okay, so in the World War II, the most common age group is around 10-year-olds. Okay, and what do you think is the most common age group now? How? 40 and 70. Give me a more precise range. 60? Okay. Anyone have any other? 40? Okay, yeah. 72. Oh, nice. I like it. It's very specific. Yeah, good. It's not going to be a whole number, so it's 58. Okay, so pretty close, right? So well, 2 years ago, it was around 58 was the most common age group, and in 10 years' time, what do you think? 72, I heard. 65? Yeah. Anyone want to go younger? No. Don't trust the fertility rate in Germany. Okay. Yeah, so it's roughly 10 years older. Okay, so 68. So definitely Germany is aging, right? So so it's something that's to be aware of, right? When looking at health conditions, right? That essentially it could be a large proportion of your sample size, right? Would be made out of older individuals, right? But well, for this one wasn't a quiz question, but what do you think for like a lower income country would be the most, like for what is it, Pakistan? What do you think is the most common age group? Much lower.
30.
Yeah. I don't know actually the exact answer, but I think it is around like 20. It's actually much younger, right? So that basically tells you it depends really on kind of, if you're looking at health conditions and so forth, it kind of depends on which countries you're looking at. Okay. All right. So reason I kind of show the age distribution, because like during that 10 years, if you look at like increase in age over that 10 years, most people will actually go through not just one, but several health conditions. Okay, so this is just a paper describing a random population cohort and looking at the types of conditions over kind of a 10-12 year period that individuals in that cohort gets, right? And eventually, you know, some of them end up, unfortunately, dead, right, due to some of those health conditions. Okay. So the top causes of death in the EU, right, may also be different, right? Because of different countries, they may have different age distributions, right? But mostly right now in Europe, the top causes are kind of heart and lung conditions, right? But one thing to just be aware of, it is different, right, for different countries. Yeah.
Different ways that death causes and death are reported to compare them.
Good. Yeah, that's an excellent question. Yeah, that's a point that I wasn't going to make. Yeah, there is yes, that simply yes. So different healthcare systems will you know, may report certain conditions, right? And
They tend to be very
oh, okay.
So
Okay.
if you're looking at heart attack data, it looks like France has got really low heart attack deaths.
Uh-huh.
But there's like specific about
Yeah. That's I did not know that. So thanks for sharing. Yeah. So, but yeah, definitely because every country will have different healthcare systems and how elderly get triaged between kind of primary, secondary, tertiary care will be different, right? I'm sure you guys remember not too long ago, the UK suffered quite a lot of deaths, right, from people with in homes, in like kind of elderly homes, and that was actually a result of how those elderly homes triage people to look at for their other conditions, not COVID related conditions, but they were triaged to hospitals where they got COVID and brought it back to the elderly homes, right? So then they kind of changed that process a little bit by how patients were referred, and that changed kind of the incidence of COVID deaths as well. So yeah, you're absolutely right that that has the healthcare system has an impact on this. Okay. So basically, there's my main point is that there's a lot of biases in how the data is collected, right? So given what we had talked about, I can define now what population health is. So population health is basically looking at health outcomes, their distribution across the population. We're looking at determinants of health. For instance, age, right, is an important determinant, and also environmental factors like pollution, infectious diseases like COVID, right, lifestyle factors, smoking, and so forth. And these will be these could be modified by policies and interventions. Okay, so like vaccines, giving out vaccines and intervention, right, that can affect some of the determinants of health, right, and also affect kind of the causal relationship between those determinants and health outcomes. Okay. So population health is kind of it's quite a kind of a multidisciplinary field, I'd say, that involves kind of analysis, data gathering, and also kind of policy work. Okay. All right. So let's see what is the distribution then of population health research across the world. Okay. So I have these two plots here. On the right is basically the number of population cohorts. So there's studies, essentially population studies. Okay. And then on the left here is number of participants of those studies and where they came from. Okay. So I'm not sure if you remember the very first question about the distribution of the population across the world. Does this reflect that distribution? Right. So yeah, so almost like 70% of the population health studies are done in North America and Europe. Right. So where some of the continents that have a very large population are kind of underrepresented here. Right. So this is something to be aware of kind of when you're working with population health data and where it comes from. Okay. So you're much more likely to find data sets coming from Europe and North America. Okay. And a specific type of population study known as like genomic association study. So this is where they also did genome sequencing of the participants, right? Because of the kind of explosion of the throughput of genome sequencing and their low cost, there has been an exponential increase in genomic data. But you can see here that the exponential increase is mainly in the European population. All right. So actually, some of the other populations are very much underrepresented, right? And what's interesting, like if you look at Africa, right, the genetic diversity in Africa between Africans is actually larger. The difference between the genetic diversity in Africa is actually larger in Africa than between Africa and Europe. okay, or Europe and East Asia. Okay? So there's basically a lot of variation we're not capturing in the data collection. Okay? So if we need models to learn on variation and association between genetic variation and variation in health outcomes, we're actually not capturing that much, despite the explosion in data.
Curious, what's the source of the quantify once we go
So there's a few genetic databases out there like dbSNP, and I think in the US and in Europe, they both host databases, and when you do a genomic study, you're encouraged to kind of deposit sequences or maybe summary statistics. So I think this plot was captured from a database called the GWAS catalog, where people published genome association studies and then deposited those summary statistics in there, and they basically counted the sample sizes for those.
Could it also be just people not posting as much from different continents on that?
Yeah, true. Yeah, there is. Yeah, we actually discussed. Wellcome actually held a workshop on this. There is some, I think, different cultures, continents. They have kind of different views about data sharing and so forth. But this we think that's only a small contributing factor, because they don't necessarily have to release the data. They just have to report they did a study on this number of individuals. Okay, so that's just like kind of a general overview about kind of just what kind of biases you should look out for in the data. So now if you do have like a population cohort, right, or a population you want to study, so how do we track changes in the health of that population, and more importantly for machine learning, how do we predict future changes? So this problem has been tackled manually, actually, without the aid of computers for quite a while. So if you're a parent, if you have the privilege of having kids, you will probably receive this booklet. So it's like a booklet that you get for each of your newborn child. And what you're supposed to do is basically plot their height and weight from birth till when they kind of get too big for you to measure, right? And basically, this is how, I think for 50 years, people essentially tracked the height and weight of children, okay? Manually plotted, and then there is actually a prediction, right? So there is basically a population mean, and then different percentiles based on your country's population, okay? So one thing to realize that this is maybe different. The distribution, the mean and standard deviation here may be different between different countries, right? But then if you plot them, and then often parents will extrapolate the curve and they do a prediction. Am I going to have a basketball star in the future or something, right? Or in this case, this is body mass index. So it's weight divided by height squared, right? So it's a measure, a very often used measure of obesity, right? So you don't want to have too high BMI, because you'll be at risk. You could be putting your child at risk of different cardiometabolic diseases later on in life, right? So we looked at that kind of data in Singapore for 994 children in Singapore, and across 994 children, we got their measures all the way till 5 years old, right? And so this is what the data looks like. So here we normalize it by the Z-score. So these are actually standard deviations from the mean, right? And so you basically on an individual level, you do get people who go up, go into 2 standard deviations, go down to 1 standard deviation. So basically become overweight, normal. Some people go underweight, normal. Not too many people go overweight and then underweight again. But there are some cases of maybe underweight and going overweight, right? So it's quite kind of noisy data, right? And then, oh, sorry, this is a bit too small. I should have enlarged it. Then on an individual level, so this is now BMI plotted by age for each individual, right? You can fit different regression models, right? So you can fit splines. This is like we're fitting a Jans Bailey is a structural equation model. And then here we're fitting a Gaussian process. So luckily T went through Gaussian processes now, so you guys should know what you're looking at now. And basically in this study was actually done in collaboration with Mauricio Alvarez here, and postdoc Arthur Leroy. And then we basically compared, we were trying to make an argument that, for this type of problem, GPs were kind of robust at fitting these curves, mainly for, I guess, two of the reasons that he highlighted, right? One is that using GPs, we're able to kind of quantify some uncertainty, right? So you can see here, like there's relatively small. There isn't like a lot of repeated measures of BMI, right? So there, and for some age periods, there may be missing recordings, right? So we show that in the red, we kind of remove some of the data points to show the effect of missing data, right? And to show that GPs may be a bit more robust to missing data, right? And then it provides the benefit of uncertainty quantification. And further, if you can extrapolate the curve, it can give you kind of a posterior for future BMIs. Okay. So maybe I'll just skip over this plot because it's about interpretation. Let's so actually outside. Okay. So what you can do, so once you fit these individuals BMI data sets, you have basically quite a few GP samples that you can then extrapolate and get a posterior probability for their chance of being overweight at 10 years of age. So in this example, for a boy and a girl, we fit GPs from data from birth to age 5, and then make a forecast of their BMI, right? So here, the purple curve is their mean trajectory, mean GP, okay? And then for each individual GP sample, we see how many of them cross this kind of threshold for being overweight, right? And from that, you can calculate posterior probability for whether they're overweight or not.
Do you fit a different GP to each individual? Some shared parameters.
Yeah, there is some shared parameters. So you can also fit a GP over the individuals, right? And also if there is some other covariates as well, you can do that. Yeah. Okay. So next, I'm just conscious about time. So but feel free to ask any questions if anything's unclear. There's just a lot of colors here, so I'm just being a bit slow. This is a kind of busy slide, but basically what we did was also, I mean, this is kind of a cartoonish slide, but we can also cluster the mean trajectories from the GPs, right? And then identify essentially groups of individuals with different BMI trajectories. Okay, that's kind of in order to slide so well. That's basically we've kind of found 5 different clusters of trajectories here. Okay, and then the interpretation of these trajectories is that you get some individuals that are kind of, I guess, born a bit large, right? And then they kind of early accelerate, so they grow, the children grow and are generally overweight. But there's also some people who born normal and stay relatively normal in their BMI, but also individuals that are born normal but accelerate, right? So they may seem normal in the first few months, but then their weight really accelerates, right? And this kind of overweight creates, being overweight at kind of infant age creates a lot of future risk as adults, right? So in particular, like hypertension, you could have like fatty liver, risk of diabetes because you have increased fasting glucose in your blood, right? So there's a lot of risk factors, right? Depending on which BMI trajectory you're on, right? But there's also a lot of influences on those trajectories, right? So everything here is still kind of association. We're really unable to infer causation here, but some of the influences, possible influences on these BMI trajectories is, for instance, your genetics, right? So there are some genetic variants associated with which trajectory you're on. Your mother, right? So your maternal behavior during pregnancy, your mother's behavior during pregnancy, how your mother feeds you, right? How you eat later on, like your diet and like your overall physiology, right? So there's a lot of confounding factors here, right? Okay, great. Yeah.
How do you go from a calcium process to this kind of interpretation? Like, what is the methodology used?
Oh, so yeah, we're just we're from these mean trajectories, we're clustering these mean trajectories, right, into these 5 groups. Okay, and then it's just interpretation of essentially then putting labels, treating these 5 groups as classes. And then you can do just association, like, just a correlation analysis or linear model. It's just traditional kind of epidemiology to do that. Good. So obesity, BMI is one large risk factor for health, population health. Another risk factor I mentioned is age, right? So age is can be pretty simple to track. So what's known as chronological age, I think most of you track that, right, by celebrating birthdays, right? So every year after your birth, you celebrate your age, right? But there's another concept called biological age, which is basically, the kind of age of your tissue, right? It could be the age of your different organs, different tissues, or even age of your brain or your mindset, okay? For instance, I may look very young, right? But I can definitely tell you, I feel like I have a back of an 80-year-old, okay? So I think I'm on the accelerated aging path right now, okay? But I'm sure some of you, may be on the decelerating age. And how many people have heard of epigenetic age? Well, a few people. Oh, great. Okay. So epigenetic age is a form of biological age. Okay. So one way to track biological age is by looking at the DNA, right? So actually, a lot of these influences like exercise, junk food, sleep, they actually create changes on your DNA. Okay. So these influences of aging will create, essentially put methyl groups on certain sites in your DNA known as CPG sites. Yeah.
Is this a correlation?
That's a good question. I feel like someone may have done an experiment. In mice, yeah. I feel like but I don't have any specific studies to quote. I can look that up for you. But there is, definitely mice studies on epigenetics, and I think especially even on brain, they measured changes in the methylation patterns from DNA collected from the brain. Yeah. So yeah, I think also even smoking, they have some studies where they definitely found like that smoking will actually induce methyl groups, additional methyl groups on the DNA. Okay.
That seems really.
Not on humans.
Okay.
Yeah, maybe we can talk about it. I'm not an experimentalist as well, so yeah, I have to say I don't have firsthand account and do no experiments, right? But yeah, I've worked with DNA methylation data, and basically, there's quite a few population studies have found that using the DNA methylation profiles, you can infer chronological age. So you can tell at least how old, given like say a piece of some skin cells and getting the DNA and getting the methylation patterns, you can actually tell how old they are, right? But that age is not actually their chronological age, it's actually their biological age, okay? So at least in older individuals, they found that the DNA methylation patterns, you can estimate the biological age of the tissue that you use to measure the DNA methylation, right? And there's quite a few what they known as clocks. They're essentially models that take DNA methylation as an input and output an estimate of age, okay? And so we looked at that actually in children at kind of at the start of aging. So that cohort which we had like BMI measures on, we also collected from their cheeks some skin cells, right? And we did methylation profiling, just 4 time points. Okay, so we modeled off of 3 time points, similar GP methods. Okay, like using multi-mean GPs to model the model from essentially 3 time points of DNA methylation. So each plot here is actually 1 DNA methylation site. Right? So there's actually for an individual, there's actually 800,000 of these plots or DNA methylation sites, right? So yeah, again, referencing back to T's lecture, there just computational resource constraints, right? Especially with building that covariance matrix with so many different sites, right? And then what we did was then extrapolate and predict the DNA methylation status at a future time point, right? Age 6. Okay, so the red dot is supposed to represent. So we look at the mean trajectory. So this is the red dot is actually the held out data point, but we can use the mean trajectory to kind of predict what the DNA methylation status is at a future time point. And then from that predicted methylation, you can use the clocks, those epigenetic clocks, to estimate the epigenetic or biological age of those children, okay, at a future time point, around 6 years. And then what we saw is actually kind of a distribution of epigenetic ages, right? So even at a very young age, where there shouldn't be too much, divergence, right, between your biological age and chronological age, we do see stuff, okay? And then you ask as an interpretation question, like what health impact that might have, right? So it's standard correlation analysis, right? Like we saw epigenetic age acceleration. So age acceleration is just the deviation between your estimated chronological age with the biological age. And we see that that's correlated with a risk factor, is diastolic blood pressure. So it's a risk factor for like hypertension later on in age. Okay, good. Any questions? All right, so I'm gonna just change path a little into a different data modality. So I've referenced some other determinants of health, mainly being kind of lifestyle behavioral determinants of health, right? And so while in the clinic and in labs, there's been like technologies advancement to kind of, you know, capture DNA high throughput and so forth. Also at home, right, there's a lot of advancement in terms of wearables, right? Like more simply like your watch, your phone. So that those devices can capture a lot of lifestyle information, right? So I'm sure some of you also have been working with wearables data. This kind of study design may be quite familiar to you. So during the start of the pandemic, we gave out, this is actually in some teaching hospitals in Sheffield, we gave out some Apple Watches, right? And enrolled health workers. So there's like nurses and doctors kind of on the front lines of the pandemic. This is like about March, April 2020, right? And then we tracked their COVID diagnoses and symptoms, as well as their physical activity from their iPhones and smartwatches, Apple Watches, through about 60 days, okay? And then we again try to, our main was to identify kind of clusters of trajectories, right? For activity data, it would be trying to identify kind of low and high activity. For COVID symptoms, there was news about, you know, long and short COVID symptoms, right? So, just to also reference, we had a collaborator at the time in Stanford, you and Ashley, whose team developed an app called My Heart Counts that was actually used before the pandemic for looking at physical activity in cardiovascular patients with cardiovascular conditions. So we kind of deployed the app because you need an app to actually collect that physical activity data. Right. And so this is for those who haven't looked at physical activity data. It's quite noisy, and there's one big problem is kind of gaps in the data. There's a lot of missingness, right? So this is, I think, from one individual. This is step count. So there's quite a few different activity measures like step count, heart rate variability, flights of stairs climbed, and so forth. But you can see here that there's like even large gaps, right, in the data. And then these large gaps are actually due to some behavioral factors. So after the study, we learned that actually for, say, some healthcare professionals like nurses, they were actually, if they're working in like surgery wards, they're not actually able to bring in, you know, phones and certain devices. So that's something we did not consider actually when designing the study. Okay. So if you have the phone and the watch, what you could do is try to impute one with the other. So this is just like a heuristic approach where we kind of train a model off of the phone data to then try to predict the watch data and then try to fill in the gaps, so forth. Right. But what's interesting is, I'm not sure if it's changed now, but when we started doing this kind of analysis, there actually isn't that many software tools for kind of data pre-processing this type of data. So later on, maybe during the break, if you have worked with this data, I would love to hear if you come across any useful tools for doing pre-processing. But I was quite surprised to find actually, you know, having worked with kind of molecular data, like genomic data and RNA, there's, you know, a lot of tools like packages in Python R for doing pre-processing quality control. But for some reason, yeah, physical activity data, there hasn't been many. Okay, so then kind of similar approaches. This one we didn't use GPs. We used linear class growth mixture models. We first, you know, smooth out the curves and then again identify clusters of trajectories, right, into kind of short and long trajectories of symptoms, right? Because the interpretation is that there are some individuals that have kind of long symptoms even after the, you know, 14 days, right? Some may have shorter symptoms. Okay. And then with physical activity data, there's different methods we tried. One of them is kind of a shape-based approach to fitting these physical activity trajectories, and then we can use kind of a k-means method to cluster those trajectories. Okay, but really, I don't really have any best practices. I'm sorry for I don't feel confident. I don't have so much experience with this kind of data to say there's some like really standard best practices, and I haven't actually seen that any real papers published on kind of best practices for this type of data. So I think this is kind of a wide open field for you guys to get into and innovate. Okay, so that was COVID. So the kind of the translational impact of that work was kind of minimal because COVID patients, the antigen test works pretty well, so they know they get COVID, and then eventually their symptoms, they're cured of COVID, right? Their symptoms disappear and so forth. So there wasn't too much utility for that modeling, but there's actually other conditions, right? Especially non-infectious chronic conditions, right? And one condition that we looked at was pulmonary hypertension. It's a kind of chronic condition of the lung and heart that has very similar symptoms as COVID, like breathlessness, tiredness. And because it has quite similar symptoms, it takes a long time actually for them to get diagnosed, okay? And even after they're diagnosed, there's actually no cure from it. So they live around 10 years or so. They've tried different treatments, and eventually they die of a heart attack. That's basically how it ends, right? So in that case, we kind of have a bit of motivation to look at the activity data before and after diagnosis and to track their trajectories, right? So this is similar data, right? So step count up here. There's other physical activity measures like pace of walking, flights climbed, and so forth, or max pace of walking. What's probably most evident of interesting patterns, so this is a kind of a mean trajectory again, is 0 is actually time of diagnosis, okay? So actually for these individuals before diagnosis, they weren't walking so much, but actually after diagnosis, they actually become more active. Okay. Oh, sorry, DC is disease control. So that's actually the COVID patients that I mentioned. So these are actually severe COVID patients. I just didn't show the trajectory here, but just their mean across that time period. Okay, so the yellow line is actually the mean of the activity of the COVID individuals, right? And then black line is healthy, right? And then and the blue line is the pulmonary hypertension, right? So the signal isn't too strong, but you can see there is some difference at least between the pulmonary hypertension and the healthy, but it's hard to kind of differentiate from the physical activity data alone, pulmonary hypertension and COVID, right? They're both kind of respiratory disease conditions, right? But what's interesting we saw is that, there is this kind of change between before diagnosis and after diagnosis, okay? Which is kind of counterintuitive why they're exercising after the diagnosis, and kind of because of we thought about it, and given the previous picture where I showed that there's actually a long journey, they actually wait a long time before diagnosis. So actually they have symptoms and they feel like there's something wrong with them, right? They're not sure before diagnosis. So a lot of patients are actually quite cautious, right, before diagnosis. And oftentimes when you receive a diagnosis, the diagnosis was kind of an intervention where they feel like, okay, I have received a diagnosis. It's a cardiorespiratory condition, and I need to now exercise to slow down my symptoms. Okay, So we didn't realize this until we kind of saw the data that actually the diagnosis itself actually induced a behavioral change that created a different trajectory in the data.
How do you find participants use the wearables before they were diagnosed?
Good question. Yeah. So a lot of them did have own smart devices, right? They just weren't consented into a study. So once we actually consent them with the app, you can pull up all the historic activity data. And some participants can go all the way back to like 2006 from there. I think maybe that's like first version iPhone or something. But yeah, I didn't know that there was even some kind of accelerometer maybe in there.
So what was the main research question of this study? Like this is just describing the statistics basically over time.
Yeah.
Specific purpose that we
Yeah, I mean the aim was basically we want to see if we can diagnose individuals earlier. Can we detect, pulmonary hypertension based on the physical activity patterns before the diagnosis? Because the diagnosis is actually quite resource consuming for the healthcare system as well. To get pulmonary hypertension diagnosis, you also need specialist respiratory physicians. They need to also do a test. The gold standard test is actually a right heart cath. They actually have to stitch a tube into the heart to measure the pressure of an artery in the heart, right? So we're thinking, okay, can we use wearables as like a non-invasive approach to detect pulmonary hypertension?
So the goal to predict the diagnosis time or the time that it might happen?
Yeah. Just try to predict which individuals in a cohort. At least right now, we're just like PH patients healthy and may disease controls and predict which ones may have the diagnosis, right? Yeah.
Do you have a sense of how much selection bias might be into the because I'm not trying to finding the participant, but obviously you're selecting the ones that have already got some kind of like step count device, for example. So do you think that could have like influenced the patterns?
Yeah, I think it would, right? But one thing I don't have an example to show this, but I think we gave out maybe like 1,000 watches. We only have really usable data for like 40 or so individuals. So actually the dropout rate is quite large, right? So
they had their own device previously, right? Because you've got data from before diagnosis.
Yeah. So it's not just there's a selection bias for people who own device, but also people who agree to share their data. I think there's also a bias there as well. Yeah. So absolutely, there is a bias, but I think these studies right now are still quite small scale. So we're kind of like we just hold any first comers basically who's willing, right? But yeah, there'll be definitely, especially I think with wearables, there's also definitely questions about equity, right? So we have like kind of a national cohort. So there's only a few PH pulmonary hypertension centers, meaning centers that can diagnose pulmonary hypertension. Hammersmith, Imperial Hammersmith Hospital is one of them. And Sheffield is another. So we basically go to those clinics, and then once they get diagnosed with PH, ask if they want to participate in the study and consent to sharing their smartwatch data. Okay. So this is kind of tackling our aim of can we predict diagnosis, right? So these are areas under ROC curves here. So we use a few different data sets. So this is just activity data from the phone, activity data from the watch. So I think there might be a mislabeling. I can't remember which one this was. What's activity data from watch? There's a few different data sets. Sorry, I should. But yeah, there's activity data from the phone and a few different subsets of sub feature sets of activity data from the watch, right? And then for the small test cohort, we're achieving like and this is not we're just differentiating. We're just predicting whether they're a PH or a healthy individual. Yeah.
So if the goal is to predict PH diagnosis, why is post-diagnosis data relevant?
So yeah, so that's interesting. So we want to see, if they have PH, if they really have PH, our hypothesis was that their physical activity level should reflect a PH, individual with PH, right? So we should actually do better at this prediction problem with data after diagnosis, right? But what we saw is that actually we can predict less accurately with data after diagnosis. And we think that's because of this, is that actually after diagnosis, the diagnosis itself changes their behavior, right? Making that data less predictive.
What's the goal of predicting after the?
So we do want to track after diagnosis. One thing is there's other interventions like treatment. So there are drugs, right? So I said there's no cure for PH. So there's a lot of drug development. There's only a few, but there's a lot of kind of investigational therapies, right? So we do want to track after diagnosis to see, especially if some people go on a clinical trial, right? Whether there's change in physical activity. Okay, so the other thing, so we knew like kind of behavioral and lifestyle kind of had an impact, right? So the other piece of information we also had from the apps was actually questionnaires about their mindset. So this is another list of a different feature set, and these are from just questionnaires that they ask. So it's more like kind of like, oh, do you feel like you're very capable of exercising? Do you feel energetic? So it's all about kind of mindset about their health and condition, right? And we find that like incorporating the activity data with these kind of mindset questionnaires was actually most predictive of diagnosis. Okay. So we also need to kind of validate, test in an independent cohort. So that was on a UK data set. So we also, like I mentioned earlier, we had the app was from a collaborator in Stanford. So they also rolled out in a similar study, not focusing on pulmonary hypertension patients, but just general variety of cardiovascular disease patients in the US, right? And then some of them were pulmonary hypertension patients. So we could do this, use that as a test set. And first time we did it, really poor performance, okay? And so it was really bad. So we were like, okay, what's wrong with the data? So we looked at some of the healthy individuals in that data set, and we recognized there's actually a distribution shift in the activity data. And our collaborators thought, that was mainly because there's actually, different US is a big place. There's different behaviors in terms of transportation, like driving. So maybe a large portion of the population are pretty sedentary. So they're not, maybe not having, many steps because the behavior, the lifestyle is different, right, in the US. So we did a little bit of transfer learning. So just incorporating some of the normal individuals in the US data into retraining our model. And then that seemed to fix the performance issue. Okay. So that's just something to kind of look out for. Again, like different countries, there'll be also like, different behaviors and lifestyle factors. Okay, good. So half an hour, so we'll just summarize here. So this is just kind of like future ambition. So we're working on collecting, lifestyle behavioral data and clinical data longitudinally, as well as kind of paired molecular data, right? And the hope is to kind of integrate these two types of trajectories to make it's kind of more personalized medicine ambition. All right, and so just to kind of summarize some take-home messages. So one is the world is getting healthier and older. Okay. Two, that the data distribution may not match the population distribution. So like some examples of there's not much data in Asia, Africa, and so forth, right? And then longitudinal data is noisy and missing, so uncertainty quantifications definitely really important. And if you're able to model and predict the future effectively, there's definitely impact on early diagnosis and treatment. And just for other kind of successful factors of success in doing modeling with this type of data, I think we need to integrate not only like molecular data, but different modalities of data from electronic health records, behavioral data from wearables. And since we're looking at older individuals, it's not just going to be one condition, right? So actually a lot of the predictive features or biomarkers, we need to look at them in multiple conditions or multiple tissues. And then finally, yeah, I'm sure people here, you guys are part of kind of interdisciplinary groups. It's a kind of collaboration, but also an iterative cycle, because you're never going to get the best performing model on the first shot. So you have to, a lot of the population cohorts are, a bit underpowered, very small. So you'll need to work with clinicians, biologists to kind of profile, to get more samples, do more profiling. Okay, I'll just stop here and take any final questions. Yeah.